Hello, IB Biology students. This is Michelle with your next installment of your video notes. Today we're going to talk about membrane transport, which is in Chapter 1, Section 4 of your IB Biology textbook. So we need to remember that our phospholipid bilayer is selectively permeable. So that idea of being selectively means that there's some control, and permeable has to do with the entry and exit of the molecules. So there's a controlled entry and exit of the molecules. Some molecules can pass through easily through a process called diffusion, or they need help, so they go through a tunnel. That would be called facilitated diffusion. But others need energy to get them through the membrane, and that's a process called active transport. Large molecules are going to need to use their own membrane to get into the cell or to leave the cell, and that's a process called endo or exocytosis. So in this section of notes, we're going to talk about all of those processes. So we need to remember that the polar phosphate heads are attracted to each other. They are charged molecules. And the nonpolar tails are going to repel any charged molecule. So it's going to prevent the passage of ions through the membrane. We need to remember that solutions consist of solutes dissolved in a solvent. So you have a solute that's dissolved in a solvent. Most of the solvents that we are looking at in biological systems are water is our solvent. A common solute is salt. So when we're looking at concentration, that's a measure of the amount of solute in the solution. So here you have two images, one pure water where there's no solute, so you can see just the blue molecules, and the other one has a salt solution with different concentrations on each side. You have 50% salt solution on the left and 20% on the right. So let's first start talking about diffusion. Diffusion is a passive net movement of molecules from regions of high concentration to low concentration. So that is the formal definition. So passive means that there's no energy required, and that's because we're going from a high concentration to a low concentration. It's like going down a sliding board. When you're at the top of the sliding board or the high concentration and you go to the bottom when you slide down, it requires no energy and it's kind of fun. Net movement of molecules, that's the overall movement. This is not that they just go from the high to the low, like going down a sliding board. But the molecules are actually moving back and forth all the time. So this is the overall movement, so overall, and then high to low, like I said, going down a concentration gradient. So diffusion can occur through a partially or selectively permeable membrane or from regions of high to low concentration in a solution or an environment. So you don't have to have that membrane separation. So here you have the 50% salt on the left, and your net movement would be to the right towards the 20%. So that you're going from the high concentration to the low concentration. So looking at this, let's look at this video and get a little bit more detailed explanation of what's going on with diffusion.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, This is one of the processes whereby materials are exchanged between a cell and its environment.
All right, so that was a little bit more detailed view of what's going on in diffusion. So here we have the higher concentration gradient leading to an increased rate of diffusion as the molecules have more energy and they'll move more quickly. So here, going from the 70% to the 20% would have a increased rate than moving from the 50% to the 20%. So that's an additional concept to gain from this idea of diffusion. All right, so the next concept is facilitated diffusion. So we still have a passive, no energy required, a net movement, so a passive net movement of the particles. We're still going from high to low concentration, but now we are going through that selectively permeable membrane with the help or facilitated by a carrier protein. So the selectively permeable membranes don't allow all molecules to pass through, and this is due to the properties of the molecules. Sometimes we need carrier proteins, also called channel proteins, that are a type of integral globular protein. So remember when we talked about the structure of the cell membrane, the integral were the proteins that went all the way through the membrane itself. And they will allow molecules to pass through. Channel proteins, though, are very specific to the molecules that they'll allow through. And this really helps with the specificity of diffusion and the selectively permeableness of the membrane itself. So they're specific to their molecules. So you have sodium channels and potassium channels that will help molecules through. And this is determined by the polar regions on the proteins. So remember when we talked about enzymes being very specific in their charge in the active site in order for the substrates to bind only with a specific substrate. It's very similar to that. With the channel protein, there's an open channel or a pore all the way through the protein, and there are specific side chains that will interact with the molecules and allow those through. So facilitated diffusion is still down a concentration gradient, still going from high to low, but so it still does not require energy. If we look at our image over here, we have a 50% solute solution. So in this particular example, we are not talking about salt. We are also not really talking about the movement of water. We're talking about the movement of the solute. So they will not be able to go through the membrane itself, and instead will need these channel or carrier proteins in order to go through. All right, so here is an example of a voltage-gated sodium channel that's used in facilitated diffusion. These are found in neurons. The properties in the outside and the inside are what allow specific molecules to pass through. Let's look at another video explanation of how facilitated diffusion works.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
All right, so another good video explaining how a process works. This time that was facilitated diffusion. So now we're going to move on to osmosis. Osmosis is still a passive net movement, but this time it's specifically of water molecules. And this is from regions of a low solute concentration to a high solute concentration. And the reverse of that would be if you have a low solute, you have a high water or solvent solution. And this is through a partially or selectively permeable membrane. So we're still requiring no energy. We're still looking at the overall movement because our molecules are moving in all directions. In this case, when we're looking at osmosis, the solutes cannot pass through the membrane due to their properties, due to their size or their charges. So osmosis is just the movement of water, or the net movement of water. So, and we always look at the movement of the water. It's typically expressed as percentages of solute solution. So in our here we're looking at 50% solute, which means low water, and 20% solute, which is high water. So our water is going to move from high water to low water. So we're still moving down the concentration gradient because we are considering the movement of the water molecules, but we express in terms of the solute solution, solute molecules. All right, so let's look at another video, this time looking at how osmosis works.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
All right, so another good video with a basic description of what is going on with osmosis. We're actually going to look at we're going to do a couple different labs looking at osmosis and the movement of water through membranes. All right, so in 2003, a gentleman named Peter Agre shared the Nobel Prize in Chemistry for his work on basically osmosis. So with the discovery of the lipid bilayer in the 1920s that we talked about in the last segment of notes, it was speculated that there was a finite degree of water permeability that would occur just by simple diffusion through the plasma membrane. However, there was a lot of work by very vigorous biophysicists and physiologists that indicated that in certain tissues, such as renal tubules and secretory glands and red blood cells, that the water permeability is much larger than could be explained through simple diffusion of the plasma membrane. So what Peter Agre's team did is they injected a complementary RNA of the protein that they had found into oocytes of some amphibian eggs. And these amphibian eggs, they're about 1 millimeter in diameter, and they were known to be quite water impermeable. So let's think about that for a minute. Why would amphibian eggs need to be impermeable to water? Okay, hope you came up with it. Amphibians, of course, lay their eggs in water, so they would need to be impermeable so that the water did not come into the egg and destroy the egg, and thus not allowing the species to rebuild. So their functional demonstration of water transport, here we have a control oocyte that is not expressing their protein, and a test oocyte here that they had injected the complementary RNA to express their protein. And this was 15 seconds after they had transferred both of these eggs, these oocytes, into a hypoosmotic culture medium. Then they let it sit, and in the lower picture, you have the same two oocytes, the control and the one expressing the protein, 3 minutes after transfer to the hypoosmotic culture medium. And you can see here that the control has had no change, but the test oocyte, the one that's expressing the protein, is swelling rapidly by osmosis and will thus begin to rupture. So what he discovered, what he and his team discovered, was a protein called aquaporin. And we now know that there is a family of aquaporin proteins that span many species. All right, so active transport uses energy. So this is another type of movement, and it is very similar to our facilitated diffusion, but this time we're using energy. The energy is in the form of ATP, and we're moving the molecules against the concentration gradient. So we're going from a low concentration to a high concentration, and that's going to use a protein pump. These molecules that require active transport, they can't pass through the membrane due to the properties and the properties of the protein. And so active transport is essential for getting this to work. It's essential, it's very key in homeostasis in organisms. It helps reset our nerves after impulse have passed through, and also helps to absorb glucose into our gut or intestinal tract. So we need that glucose that we eat. It is in a high concentration when we eat it. It needs to go into the low concentration of our intestinal tissue so that we can absorb it and thus use it. All right, so here's an animation looking at secondary active transport. So this is a type of active transport. So we have an enzyme here. How do we know it's an enzyme? Oh, it ends in ASE. So this is a sodium potassium ATPase. And this is a sodium glucose symport. So you can see here this molecule is gathering sodium on one side and potassium on another. The ATP comes in. It releases the energy by breaking into ADP and a free phosphate ion here. And now we have a buildup of sodium. So sodium can go to the symport and thus pull the glucose in. So that was an example of active transport, the active transport of switching the sodium and the potassium molecules and then getting that buildup of sodium working with the glucose to pull that in. All right, so remember that these protein pumps, again, are specific to their molecules, and the molecule binds to the active site of the pump, so very similar to an enzyme. And then the release of ATP is going to result in a conformational change in the shape of the protein pump, which will allow the molecule to be pushed to the opposite side of the membrane. So let's watch another video. This one is specifically how that sodium-potassium exchange pump works. I also have models of this that we will use for a hands-on demonstration in the class as well.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So again, we'll be looking at that particular pump a little bit more detail in class when we get to our hands-on activities. All right, so the last concepts that we need to look at are the idea of exocytosis versus endocytosis. So the prefix exo does mean to exit, cyto means cell, and is or cis means process. And so exocytosis is a process of exiting the cell. An endo prefix means entering, so endocytosis is a process of entering the cell. So exocytosis is the export of macromolecules from the cell, and endocytosis is the import. There are two different types of endocytosis. You have phagocytosis, which is cell eating. So this is the ingestion of molecules using pseudopods to surround the food and form a vesicle. And then you have Pinocytosis, which is typically referred to cell drinking, and this is the ingestion of molecules forming a vesicle at the surface of the membrane. So let's look at videos of both of these because they are slightly different. All right, so here we're going to try and eat this molecule, forms the phagocytic vacuole, and then it combines with the lysosome so the lysosome can break it down. Watching it one more time. So these are those pseudopod arms that connect, encapsulate the molecule, and bring it into the cell. All right, let's look at the other one. Pinocytosis. So here's pinocytosis. We have a solute. And this one's a little different in that the pseudopods don't reach out. Instead, the cell membrane dips down and the molecules come with it. So slightly different. And in this case, the molecules are so small that they do not need to be broken down by a lysosome. They can just leave the vesicle. All right, we're going to do more exploration with exocytosis and endocytosis as well in class, some hands-on activities to really understand this a little bit more. But we have had some information. In 2002, we actually figured out the method for a membrane fusion. And this was discovered by Yang and Huang using X-ray diffraction methods. So what we were able to find out is when the vesicle approaches the plasma membrane, we do know that all membranes are made of a phospholipid bilayer. So there's some properties that are shared and common between the two. And what happens is here, the membranes begin to fuse. So remember the fluidity of the plasma membrane, that those phospholipids can flow around each other. And for a moment, they pinch in and there's a single bilayer between the two structures at the point of contact. And this is a temporary intermediate structure. And then the membrane pore opens, so this area here pinches off as they are touching, and the contents can pass through. Remember, though, throughout this whole process here, there is never an unbroken section of the bilayer. Okay, so even if you're looking at this zoomed in section here, it appears to be broken, but we need to look at the greater picture of these membranes here being connected. So this is almost one cell as those membranes are connected. All right, so that's the end of our membrane transport notes. Thank you for watching. Remember to bring your notes to class as well as your questions. See you in class.